At the initial stage of eukaryogenesis, groups of interconnected microbes, bacteria and archaea, formed communities. These included various types of microorganisms such as Clostridia, Actinomycetes, Planktomycetes, Geobacteria, and Ascardarchaea. The microbes existed in a synergistic syncytial relationship with one another, forming syntrophic consortia. Such microbial communities are still commonly found in nature today. About 2.5 billion years ago, a consortium of microbes was infected by viruses, which led to the lysis of a significant part of the microbes integrated into the community. The surviving microbes formed new consortia. Here is how the archaea functioned before the viral invasion. Genetic information from DNA was transcribed into RNA, which then moved to the ribosomes. There, the RNA sequence was translated into proteins that carried out vital cellular functions. Here is the function of the archaea after the virus invasion. These viruses belong to the retrovirus group. After infection, the genetic information of the virus was integrated into the genomes of the microbes. Intronic inserts appeared in the genomes. Accordingly, the RNA and proteins synthesized were defective and caused the lysis of the infected microbes. It became necessary to cut out viral introns and create the appropriate apparatus for this. A similar mechanism, in the form of a spliceosomal apparatus, already existed in archaea in a primitive form. Some archaea, let's call them smart archaea, managed to perfect this apparatus and adapt it to the created reality. Bacterial RNA was capped in a similar way to viral RNA and was exported outside the archaea. At the same time, the secretory systems of archaea were transformed into cellular pores. Portals and eukaryotic type ribosomes were created. Archaea may have received eukaryotic type portals from Planktomycetes. In this way, the proto-nucleus was formed from the central archaea. These changes allowed the messenger RNA and ribosomes to be transported from the archaea to the extracellular space of the microbial community. Transcription remained in the nucleus, while translation moved to the extra-archaeal space. As a result of this reorganization, the archaea were able to avoid the existential threats posed by intronic viruses, ensuring the integrity of their genetic material and maintaining the necessary for survival. The same issues later arose in bacteria as a result of viral infections. The integration of viral elements into the microbial genome and the subsequent synthesis of defective proteins led to the lysis and destruction of bacterial cells. Since bacteria lacked a spliceosomal apparatus, ubiquitin system, and other mechanisms necessary to combat viral infections, they adopted an alternative solution. They transferred their genomes to so-called smart archaea by processing and splicing genetic material within the proto-nucleus. The bacterial genome RNA was purified and freed from viral introns. After this, the bacterial messenger RNA synthesized within the archaea was transported into the intercellular space of the community. In the process of evolution, bacterial cells that were emptied of their genome progressively lost their structure and formed cistern-like shapes resembling endoplasmic reticulum. Eukaryotic type ribosomes gradually positioned themselves on the surface of the deflated bacterial cell membrane, which led to the formation of the rough endoplasmic reticulum. Later, here transferred the translation of bacterial and other messenger RNAs. The synthesized proteins reached the inside of the endoplasmic reticulum through portals present in the endoplasmic membrane. The flagellated bacteria, probably Clostridia, included in the community of cells, were transformed into the smooth endoplasmic reticulum as a result of deflating and parallel gene transfer in the smart archaea. And, as a result of the parallel transfer of genes from bacteria to archaea and deflation of the cells, the Golgi apparatus arose. From geobacteria, the central archaea received the genetic apparatus necessary for the synthesis of sterols. Here we see how ribosomes are attached to the surface of the rough endoplasmic reticulum. Messenger RNA, which comes from the archaea, moves to the rough endoplasmic reticulum, where it is translated into proteins. Once synthesized, these proteins enter the endoplasmic reticulum, where they undergo folding and initial modifications. This process ensures that the proteins are properly folded and ready to function within the cell or be transported elsewhere in the microbial community. In the smooth endoplasmic reticulum, vacuoles begin to form and are released into the cytosol. The Golgi apparatus began to form lysosomes. As a result of the parallel transfer of part of the genes of the alpha proteobacteria included in the community to the central archaea, mitochondria, and chloroplasts arose. At the same time, phagocytosis arose, presumably bacterial type, protophagocytosis. Let's call it endocytosis, had played an important role at other stages of eukaryogenesis. However, it is impossible to reconstruct the exact chronological sequence of individual stages. Endocytosis may be used for the directed transport of solid substances, including drugs, into cells, and in the treatment of various diseases, including cancer at the periphery, presumably through the fusion of Asgard archaea and geobac. Teria, the proto-nucleus, along with the endoplasmic reticulum, Golgi apparatus and mitochondria, was internalized into the syncytium of the fused microbes. The transfer of genes from Asgard archaea and geobacteria to the proto-nucleus and the elimination of their genomes led to the formation of the cytosol and eukaryotic membrane. Sterols imparted elasticity to the membranes and actins imparted motility. The central archaea possessed flagella, which they used for locomotion and cell division during eukaryogenesis. These flagella gradually transformed into centrioles, structures that subsequently played a crucial role in organizing the cell's microtubules, and mitotic division in eukaryotic cells.